Mycobacterium tuberculosis. What is Mycobacterium tuberculosis? It is the bacterium that causes tuberculosis. It is also known as the TB. TB is a contagious infectious disease that primarily affects the lungs but can also affect other parts of the body, such as the kidneys, spine, and brain. As you can see in this figure, there is a human body in which the Mycobacterium tuberculosis colonized in the lungs. Mycobacterium tuberculosis mainly identifies in the lungs as well as in the trachea or in the other parts of the human body such as the kidney, spines, and brain. So these are those parts of the human body, these are those organs which are mainly affected by the Mycobacterium tuberculosis. In the next slide, you can see there is the transmission between the two persons. A one infected person, when it is coughing or sneeze, its respiratory droplets carry the Mycobacterium tuberculosis and use the portal of exit and transmit the bacteria from one person to another. As you can see in this figure, the bacterium is easily transferred from the respiratory droplets from one person to another. So this is the route of transmission of Mycobacterium tuberculosis. In the next slide, when a person is infected by the TB, then what are the symptoms? The TB is characterized by the symptoms such as a persistent cough, sometimes with blood sputum, fatigue, weight loss, night sweats, fever. These all are the symptoms which are relevant to the Mycobacterium tuberculosis. But the cough with the blood sputum is one of the specific symptoms which are mainly relevant to the TB. It can be very serious and potentially life-threatening if it left untreated. So, as you can see in this below figure, there is a blood sputum image, which is showing a specific symptom of the Mycobacterium tuberculosis. So, how the blood sputum looks like you can see in this figure. In the next slide, there is a structure of the tuberculosis. As we all know that Mycobacterium tuberculosis is the bacteria which cause severe infections in humans. Then how it looks like. This is the picture of the Mycobacterium tuberculosis under the microscope, as you can see in this diagram. How this Mycobacterium tuberculosis looks under the microscope. These are the rod-shaped bacilli which is representing as a TB. So, if we are going to the anatomy of the Mycobacterium tuberculosis, we are going to analyze the structure of it. As you can see, this is the rod-shaped bacteria in which there is a flagella. Flagella are mostly extended from the interior side of the bacteria body. After that, there is the plasmid, ribosomes, mesosomes, plasma membrane which lie under the capsule. Then there is a DNA which is the genetic material of the Mycobacterium tuberculosis. Then there is the cytoplasms, capsule, and pili. There is the two main terms, mesosomes and ribosomes. Ribosomes are the cellular structures which are mainly responsible for the protein synthesis, and it can be found in the cytoplasm of a prokaryotic and eukaryotic cells. They read the messenger RNA and assemble the amino acids into protein. So this is the function of the ribosomes. Likewise, the mesosomes on the other hand are specific structure found in some bacterial cells, just like the Mycobacterium tuberculosis. They are invaginations of the plasma membrane and are mainly involved in the process like cell division and DNA replication. So this is the function of the mesosomes. There is a different factors which are mainly involved in the structure of the Mycobacterium tuberculosis, as you can see in this figure. But every factor has some specific function. So after that, you can see there is the genetic material of the Mycobacterium tuberculosis is the DNA, and in the bracket it is written as nucleoid. Nucleoid is a region where within the prokaryotic cells such as the bacteria where the genetic material DNA is located. Prokaryotes lack a true membrane bounded nucleus, so the DNA is present in a concentrated area called the nucleoid. So this is the function of the nucleoid, how the nucleoids perform its function. After that, there is a word about the plasmid. A plasmid is a small circular double-stranded DNA molecule that can exist independently of the chromosomal DNA in bacteria and some other organisms like archaea and yeast. Plasmids are not essentially for the cell survival, but they can carry some important genes that can provide advantage to the host cell under certain conditions. So, this is one of the main factor of the bacteria structure. This all about the mycobacterium structure, and now we are going towards the next slide. So the next slide is about the sample collection box and the identification of sputum. So it is mainly referred to a question which is how to collect sample of TB, means Mycobacterium tuberculosis, and identify the sputum. So as you can see, there is a sample collection box which are mainly used for the collection of the TB samples. It is looks like this, as you can see in this picture. And after that, in the next picture, you can identify about the sputum and the saliva. What's the difference between the sputum and what's the difference between the saliva? The sputum is the thick and sticky, while the saliva is the thin and the watery. So for the diagnosis of the Mycobacterium tuberculosis, we mainly prefer the sputum, which is thick and sticky. So collecting a sputum sample for the identification of tuberculosis is an essential step in diagnosing the disease. Firstly, proper precautions are one of the most important steps. Then the patient preparation, and then the sample collection. This is the sample collection. Collect the sputum sample in a sterile leak-proof container. Looks like this. The patient may need to provide multiple samples for more accurate diagnosis, especially if they are suspected to having TB. And after that, after the collection of the sample, the next one of the major step is the labeling. Label each container with the patient's name, unique identifier, date, and time of collection to ensure proper identification and avoid mixing up during processing. And after that, when we label the sample collection box, it is ready to proceed to the medical laboratory. Then there is a step of the transportation. Transport the sputum sample to the laboratory for testing. If there is a delay, store the sample in a refrigerator at a suitable temperature. And after that, when the sample is safely delivered to the laboratory, then the next step is about the laboratory testing. As you can see, then this step will become identification of sputum, laboratory testing. In the sputum sample will be analyzed by the various methods such as the smear microscopy, culture, and the other tests also. We are going to discuss it one by one in the next slides. Now this slide is about the diagnosis of Mycobacterium tuberculosis. The first and the most common test which are going to perform in order to diagnose it is the sputum smear test. This test involves examining the sputum under a microscope to detect the acid-fast bacilli including Mycobacterium tuberculosis. So this test is also known as the smear microscopy. In this method, the sputum is spread on a slide and then it is stained and examined under a microscope. Acid-fast staining is commonly used to detect the characteristic acid-fast bacilli of Mycobacterium tuberculosis. The main staining which are going to use in the sputum smear test is the acid-fast staining, and the results look like in this figure. As you can see in this figure, these are different Mycobacterium tuberculosis are shown, which are going to be appear in a red rod-like structure. After this, the another test is about the tuberculin skin test, also known as the Mantox test, involving injecting a small amount of purified protein derivative tuberculin into the forearm and assessing the reaction after 48 to 72 hours. This test is also used to diagnose the mycobacterium. A positive results indicate exposure to the TB bacteria but does not confirm the active disease. So this test is also performed in order to diagnose the mycobacterium. So it is also known as the one of the most important diagnostic test which is going to explore the bacteria. which causing the tuberculosis. It involves injecting a small amount of purified protein, as you can see in here, PPD, from the TB bacteria under the skin, usually on the forearm. After 48 to 70 hours, a healthcare professional examines the injection site for a raised red dump called an induration, as you can see in this figure, how its results look like. If it is positive, then it is appear like this. The size of the induration helps determine if the person have been exposed to TB or has a latent TB infection. So this is the sign of the positive Mentox text. And in the next figure, you can see how it is going to be performed. There is a healthcare provider who is going to inject the purified protein derivative into the forearm of the patient in order to diagnose the TB infection. The next one is the culturing of sputum. Culturing a sample of sputum or other body fluids in a laboratory allow for growth and identification of Mycobacterium tuberculosis. So in a culturing step, there is a various different process work steps are followed. Such number one is the sample collection, as we can discuss it earlier. Then there is the decontamination process. Before culturing, the sputum sample undergoes a decontamination process to remove any contaminant or the normal bacteria that may interfere within the growth of the Mycobacterium tuberculosis. And after the decontamination, there is a step of inoculation. The decontaminated sputum is then inoculated into the specific culture media that supports the growth of the TB bacteria. The most common used medium for the TB is the Lowinston Jensen culture, as you can see in this figure. And after the culturing of the sputum for the Mycobacterium tuberculosis, it is incubated under the usually 37 degrees Celsius for several weeks. Mycobacterium tuberculosis is a slow growing bacteria and it takes up to 6 to 8 weeks for visible clunies to appear. So it is a very it is a very time duration test also, because the culturing takes so many weeks, because the bacteria is a very having a very slow growth. And after the clunies are going to appear in the culture media, we are going to examine these clunies. After incubation, trained laboratory scientists examine the culture for the presence of characteristic clunies of Mycobacterium tuberculosis. The clunies have a rough, as you can see in this figure, how the clunies are grow. The clunies have a rough, dry, and bulbed colored appearance, as you can see in this figure. This is how the clunies of the Mycobacterium tuberculosis looks like. So this is the steps of the culturing of sputum. And culturing of sputum and the smear microscopy, these two tests are mostly common and a routine diagnostic test for the diagnosis of the TB infection in patients. So after that, after the diagnosis, there is a treatment. The treatment of tuberculosis typically involve a combination of multiple antibiotics taken over an extended period of time. There is a first line medications which are also involved in the treatment of the TB infections, as well as there is a sometimes second line medications are also going to be involved in it. But here we are just going to discuss about the first line medications, which is about the isoniazid, rifampin, pyrazinamide, ethambutol, streptomycin. Injection for initial phase. These are those treatment. These are those antibiotics which are given to the patients when the patient is going to expose with the TB infections. And after that, the prevention strategies is one of the major and important tasks. We should have to prevent ourselves from the infected patients through wearing the mask, by performing the hand hygiene practices on our daily routine, and by maintaining a social distancing, because Mycobacterium tuberculosis is the bacterium that easily transmits through a respiratory droplet from one infected person to the another one. So prevention strategies is one of the most important part in order to prevent ourselves and the whole community, whole the populations from the infectious disease, and it is one of the major important factor in order to break the chain of the infection from spreadness.